All right, so let's take a look at this figure over here on your right-hand side. The stem cell that starts all of your blood cells is actually called a hemocytoblast, okay? This can differentiate into 5 different cells: the proerythroblast, the myeloblast, the lymphoblast, the monoblast, and the megakaryoblast. Okay, so again, stem cells can become multiple things. In this case, this specific stem cell can become these 5 cells. Now, the proerythroblast over here will become the early erythroblast. Notice that it has a nucleus. Then it kind of starts condensing down and becomes the intermediate erythroblast until it becomes the late erythroblast. Notice the nucleus, notice the difference in size in the cell, and notice that the shape is changing, okay? By the late erythroblast stage, eventually it spits out its nucleus and becomes a reticulocyte. This is the last phase before it actually becomes an active red blood cell. Okay, notice the shape of the red blood cell. It is very different than this globule shape up here. And we're going to talk about why that happens in just a minute. But for right now, I want you to see kind of that evolution of that cell. Now, the myeloblast. All of your granulocytes, your white blood cells that are granulocytes, are going to come from this myeloblast. You've got the progranulocyte here, pro meaning kind of like the before, and it becomes a basophilic myelocyte, an eosinophilic myelocyte, and a neutrophilic myelocyte. Notice that all of these names are close to what the granulocytes are actually called. So as they evolve, eventually you get the band cells, the basophilic band cell, the eosinophilic band cell, the neutrophilic band cell. I was going to say bland cell. That didn't work. The next step is the final one. You get the basophil, the eosinophil, and the neutrophil. These are your granulocytes. These are your white blood cells that are granulocytes. Okay, here's white blood cells. Now, the next two, the lymphoblast and the monoblast, these are going to be your agranulocytes, and they are specific to what they will become. The lymphoblast becomes the lymphocyte. The monoblast becomes the monocyte. So these are your agranulocyte white blood cells. Okay? Now remember what I said about the thrombocyte. I said that's a really bad name for it because it's not a cell. So this stem cell, the hemocytoblast, can also become this megakaryoblast. What does mega mean? Big, right? So eventually this becomes a megakaryocyte. And that cell, this one, starts to break apart. So if you look here at the base, you'll see pieces of it being pinched off. These are the platelets. Notice that they're not actually a cell. It's just a chunk of this megakaryocyte. That's one of the reasons why I say that it's not a good way of naming that. Hello, thank you. So let's talk red blood cells. Red blood cells have a very specific shape to them. You can see it here. They are a biconcave disc with thicker edges than they have in the center. You can see that it's thicker out here than it is in here. And they are flat, right? They're not round. They're not globular. They don't look like a sphere. They actually look relatively flat. That actually allows for greater surface area, and it makes movement of gases into this cell a lot more rapid. Okay. It allows for easier bending and folding as well. That means that I can decrease the size of this cell and allow it to pass more easily through blood vessels because I'm capable of folding it up like a tortilla instead of having something that acts like a cork because it's a big old sphere. The original cell loses its nucleus, which you saw in the previous slide, and almost all of the organelles when it's mature. The main component within the red blood cells is something called hemoglobin. This dude is super important, okay? It is a red pigment protein, and it fills about 1/3 of the volume of this cell. Hemoglobin is what carries our oxygen. So having it and having it in spades is really important. Minor components include things like lipids, ATP, and the enzyme carbonic anhydrase. Remember, I told you that we could actually take carbon dioxide and make it into a buffer? This is the enzyme that does that. The carbonic anhydrase is the thing that can convert carbon dioxide into our carbonic acid, which then becomes our bicarbonate ion. So if you have a cell that is a sphere like this, okay, and you have stuff inside of that cell that needs to carry oxygen, that oxygen has to traverse that entire distance to get into the center of this beach ball, okay? Again, if we're talking about a cell that's that classic circular shape, that's a really long distance to travel. However, if you have a cell that is shaped like this and it needs to get to the center, this distance here is a lot smaller than this distance here. So having this flat cell gives you greater access to the surface of this cell and to the inside because it is as flat as it is. Excuse me. That means that getting inside of something this shape versus something this shape is a lot faster. If I'm an oxygen molecule, I don't have very far to travel. Hang on one second. I don't have very far to travel to get to the inside of this cell here versus a sphere where I've got all of this space that I'm going to have to traverse to get into this cell. Also, if you've ever seen anybody make a tortilla, right? When they first make it, they make a ball and then they roll it out, right? How much surface is on the ball? Not much, but when I actually roll that ball into the shape of a tortilla, then you actually have a lot of surface that's available, right? It's the same concept. If I take this cell and flatten it out, then I get a lot more surface available to me than if I kept it as a sphere. So the primary function of oxygen, the primary function is oxygen transport. This is my primary function. This is what I am doing as a red blood cell. That's what I was designed to do. I take oxygen from the lungs into the body tissues, whether that's the fingers, the toes, the top of the head, the pecs, the gluteus maximus, it doesn't matter. That's my job. 98.5% of oxygen is actually linked to the hemoglobin that is inside of your red blood cell. Only about 1.5% of that oxygen is going to be dissolved in the liquid plasma of our blood. It can also take the CO2 waste, which I mentioned before, from the body tissues to the lungs so that you can go and breathe it out. Okay? Red blood cells eventually will rupture, and when that happens, we call it hemolysis. Okay? Hemo, because of the hemoglobin, lysis means to break. Hemoglobin, in order to function properly, actually has to be inside the cell. The minute you take it out of the cell, it unravels and it won't work anymore. It basically denatures. So I always describe denaturing of a protein as having a cable knit sweater and you find a thread, so you start yanking on it and it unravels the sweater. Are you going to be able to re-knit that sweater? Probably not. I crochet, and believe it or not, I knit, and I know I wouldn't be able to do it. So when a protein denatures, it falls apart. If that sweater, you keep pulling on it and eventually it's just a bundle of yarn, are you going to be able to use it as a sweater anymore? No. So when hemoglobin denatures, once it's outside of the red blood cell, it doesn't work anymore either. Now, the CO2, the carbon dioxide we're talking about transporting, we transport it in three major ways. About 7% actually dissolves in the plasma. So it's a little bit easier to get it to dissolve in the plasma. 23%, though, is attached to hemoglobin. That leaves a buttload of extra, right? Because if we're talking 100%, this is only 30. Where's the other 70%? It's here. Remember I told you about that carbonic anhydrase being present and it can convert it into a buffer? 70% of our carbon dioxide that we're picking up actually gets turned into the bicarbonate ion, our buffer that is in our blood that helps us to maintain our pH. So the carbonic anhydrase, which is that enzyme I mentioned, is the enzyme responsible for taking carbon dioxide and water and converting it into carbonic anhydrase. Carbonic acid. My bad. Carbonic acid is an acid, which automatically means if you put it in solution, it splits apart into a hydrogen ion and something else. In this case, it's the hydrogen ion and the bicarbonate ion, which is our buffer that we use to transport the CO2 in the blood. When we get to the lungs, we basically just do a reversible reaction and we blow out the carbon dioxide. So we use carbonic anhydrase to turn this bicarbonate ion and hydrogen ions back into carbon dioxide and water. Okay? So let's talk hemoglobin. This is hemoglobin. You can see that I've got 4 squigglies, one orange here, one orange here. So beta 1, beta 2, alpha 1, and alpha 2, right? Those are polypeptide chains. It's chains of amino acids, kind of like a pearl necklace, that fold into that specific structure that you are seeing. I need 4 of them to make a hemoglobin. As well, you will notice that I've got this blue. It almost looks like a coin in the middle of each of these. Yes? That is this over here. This is the heme group. So the squigglies are called globin. This is the heme, and it is heme, not hemi, because it's hemoglobin, so it's heme, just FYI. Because I've heard people say hemi, and I'm like, okay, that has to do with Dodge trucks. That doesn't have to do with blood. So each polypeptide chain, the globin, okay, is bound to one heme. So you've got 4 chains, you've got 4 hemes. There are 9 hemoglobin types based on the amino acid sequence that is um, in these little squigglies that you see on the upper left-hand side. So you've got alpha, beta, gamma, delta, and embryonic, as well as fetal. But for the most part, if you're looking for what's the most predominant ones we find, it's the alpha and the beta. We usually have 2 alpha, just like you see in the picture here, and 2 beta, just like you see in the picture here. Okay? The heme itself, because it contains iron, is actually going to give the red pigment that we associate with blood. This is the thing that does that. Okay, this heme group. Now, throughout our lifetime, we actually have 3 types of hemoglobin that exist, and each one has a different affinity for oxygen. So what's affinity mean? And it's not forever, that's infinity. Affinity means liking for or attraction to. Okay, I have a very high affinity for Jensen Eccles, right? So affinity is how much it likes or is attracted to oxygen. We have embryonic hemoglobin, we have fetal hemoglobin, and we have adult hemoglobin. Okay, and each one has a different affinity for oxygen. Where does baby get everything that it gets when it's in mom? It takes it from mom, right? That's literally the only way that I can get anything because I'm not outside of the womb yet. What that ultimately means is if I want oxygen, I have to steal it from mom. Ultimately, that means that embryonic and fetal hemoglobin, see what color I have, these two have to have a higher affinity, a higher attraction for oxygen than adult. Why? Because I have to take it from mom. If I'm weaker, I'm never going to get any oxygen. So embryonic hemoglobin is about the first trimester, about the first 3 months of development, and it's got a very high affinity for oxygen. So when mom's blood goes by, I take, okay? Fetal hemoglobin is what's going to replace the embryonic about the 3rd month. And again, fetal has a stronger attraction for that oxygen than adult. So I'm still, as a baby, still in mama's womb, able to steal that oxygen from mom. This is also one of the reasons why mothers tend to get extra blood cells made, extra red blood cells made, and they can suffer from high blood pressure, preeclampsia, because they're basically trying to keep enough oxygen for themselves while baby's stealing it. Now, the adult hemoglobin, by birth, baby has about 60 to 90% adult hemoglobin, but it's not until about the ages of 2 to 4 that their entire body is now just adult hemoglobin. Now, remember, by this point, they're actually out in the atmosphere and they can go all by themselves and don't need to take anything from mom. So having adult hemoglobin makes sense at that point. Now, let's go through the numbers real quick. Each red blood cell, each one of these little cells right here, contains 270 million hemoglobin proteins. 270 million of those in each one of these. And each one of these is carrying 4 heme groups, right? Meaning there's 1 oxygen here, there's 1 oxygen here, there's 1 oxygen here, and there's 1 oxygen there. Okay? At any given time, we have between 20 and 30 trillion red blood cells in our body. How many oxygen are we holding at any given time? Looking at those numbers. Wow, right? I mean, we're carrying tons of oxygen, which is fantastic for us because we need it. But if you look at the numbers, 20 to 30 trillion red blood cells that have 270 million hemoglobin proteins each, and each one of those hemoglobin proteins is capable of carrying 4 oxygen. That's a lot. 25% of all of the body cells we have are red blood cells. We have 100 trillion in total. That's a ton of oxygen carrying right there. So, iron is required for normal hemoglobin function because oxygen binds to this iron right here that's in the center of the heme. Okay? Where do we get it? Well, usually we get it through our diet. If you eat any type of meat, you're getting iron. If you eat green vegetables, you're getting iron. If you are a vegan, this could be a problem because if you're not eating meat, the amount of iron you get from green vegetables is not near as much. So if you're a vegan, you have to be aware of that and make sure that you're altering your diet to either include an iron supplement or so that your vegetable intake still gives you enough iron. Exposure of oxygen to oxygen binds 1 oxygen to each of the heme groups. Okay? When you do that, when oxygen and heme are bound together, you call it oxyhemoglobin because it's hemoglobin with oxygen. Without it, it's called deoxyhemoglobin because it's not there anymore. The amino acids of the globin are actually what bind to carbon dioxide. So carbon dioxide does not bind to this guy. Carbon dioxide binds to these guys here, the little squigglies. So when that happens, when instead of carrying oxygen, you're carrying carbon dioxide, we call that carb amino hemoglobin. It is also capable of bonding to nitric oxide, which functions as a chemical signal in the body, kind of like a hormone, and it actually induces relaxation of smooth muscle. We think this has yet to be confirmed, but we think it might play a role in blood pressure because if nitric oxide is present, the smooth muscle in our arteries and veins relaxes, which means that it would drop our blood pressure. It would be able to bring our blood pressure down. So, red blood cell production. Negative feedback loop. We've got homeostasis, we've got our window. When we get down here below that window, though, we need to get back up into the window, right? That's negative feedback. Negative feedback decreases the amount of deviation from homeostasis. So negative feedback goes, get back to where you were. So in a negative feedback loop, the thing that starts this is a decrease in blood oxygen levels. In other words, I don't have enough red blood cells to carry enough oxygen. The first organ to notice this is going to be the kidney. The kidney has your entire blood volume going through it 70 to 80 times a day. It's going to notice really, really fast if there isn't enough oxygen in there. So when that happens, when the kidney senses, there's not enough oxygen, it releases a chemical called erythropoietin. Okay, so it releases a chemical messenger called erythropoietin. Erythropoietin travels to the red bone marrow and says, hey, dude, we don't have enough red blood cells. You need to start making more. So the red bone marrow starts to increase the amount of red blood cells being produced. What happens then? Well, we start seeing an increase in blood oxygen levels. When this oxygen-rich blood comes back to the kidney, this is no longer happening. So the kidney stops doing this because I have plenty of oxygen now. So this process is how we, one, know we need more red blood cells, and two, how we get them when we realize that we don't have enough. So, red blood cells have a finite lifespan. The only way for a red blood cell to, the only way for a cell to survive and fix things is for it to have a nucleus that has the instructions on how to fix things. Red blood cells don't have that anymore. Remember, they spit it out. So ultimately what that means is they don't live for very long. In women, they live for about 110 days. In men, it's about 120. And believe it or not, that difference has to do with testosterone levels. So, as I said, without a nucleus, without a nuclei, they have no way to produce new proteins or divide existing proteins or enzymes. The plasma membrane components that are there and other structures will start to break down. And since I don't have the instruction manual on how to make new anymore, because I spit out the nucleus, once they start to break down, that's it. They're done. It's broken down. So ultimately, as time goes on, they become less stable. They become less able to transport oxygen. The plasma membrane will eventually start to break down too and become more fragile. And they can rupture. And when they rupture, they will release hemoglobin. So what do? Well, age, damaged, or abnormal red blood cells get taken to the spleen, the liver, and some other lymphatic tissue. And the macrophages, macro, big, phago, eat, the macrophages actually isolate the hemoglobin. They actually take it back. Your body is the ultimate recycling center, and I'm not going to let that iron just get taken out of my body if I can avoid it. Why? Because I need it. You don't have oxygen, you die. So,